Ka tangita titi, ka tangita kaka, ka tangi hoki a hau, ti he Māori ora. E na mana e na rea a ra rakatera mā tēnā koutou, tēnā koutou, tēnā rā kātou katoa. Good evening and welcome to this, the University of Otago Rowheath Trust Award and Carl Smith Medal Lecture to be presented by Dr Karl Iremonger of the Department of Physiology. My name is Richard Blakey. I'm the Deputy Vice-Chancellor for Research and Enterprise here at the University of Otago, and I'm here with an academic, that's the ones of us in the silly robes, consisting of Associate Professor Fiona McDonald, Head of Department, tēnā koe, Professor Vernon Ward, Dean of Biomedical Sciences, tēnā koe, Professor Paul Brunton, Pro Vice-Chancellor Health Sciences, tēnā koe, DVC's Professor Pat Craig and Helen Nicholson, tēnā korua, and our Vice-Chancellor Professor Harleen Hain, tēnā koe o te tumoaki te wharewana o Otago. Look, and it's my great pleasure to share this celebration of outstanding research achievement with you all, friends and whānau of our medal recipient, members of the university community, citizens of Dunedin and the region, and there may even be some representatives of the media. No mai hara mai welcome. And a particularly warm welcome and greeting goes to Carl's supporters and family who are here tonight, with greetings to Carl's mother and father, Gail and Andrew, wife Jo and daughter Sylvie, tēnā koutou to you all. Look, this is one of the most enjoyable duties that I have the privilege to perform in my role every year, and it's really nice to be able to do this in an official capacity tonight, although really what I'm interested in, as many of you are, is to hear the details of the talk tonight. It's both a ceremonial element but also professional and academic stimulation for us. Now, to give you an idea of the way things that will proceed tonight, the order of ceremonies is that I will say some words and then invite Associate Professor McDonald to introduce the lecture. Following the lecture, I summarise the significance and give a vote of thanks. And then, provided the lecture is suitably impressive, and I have warned Carl that this is a test as well, we as a committee decide the award, but you as the public should affirm that. We will present Carl with his award and medal. And finally, and then very importantly, we will retire to a reception in the staff club for refreshments and fellowship. But first I thought it's quite useful. This is a Carl Smith Medal and Rowheath Trust Award. Some background to the name of this medal and award. The purpose of these, which began in 2003 and are always awarded together, so you can't get one without the other, is very clear. They are to, quote, promote research at the University of Otago and give recognition to outstanding research performed of early career staff, which is for those who have been at Otago for at least 2 years, but have a total research career appointment of at least 10 years. It's research experience and age that is not the defining factor, so that 10-year limit is what we use here to define early career research. Now, this year we had 8 outstanding nominees who all pretty much met the award criteria with bells on, and after much painful deliberation, we chose Karl as a recipient. What stood out to the committee about Karl's nomination was not just the major internationally significant achievement he has already made, and we'll hear about tonight, in, amongst other things, the discovery of a new type of neural structure, I love it, called the dendron. As I understand it, it's a hybrid between axons and dendrites. But as well as that, it's the breadth and depth of your contribution to the understanding of the brain, the development and implementations of new techniques here at Otago, and your outstanding acute communication abilities that led us to that decision. Fiona will outline many of your achievements, I'm sure, but what really helped the case, in addition to the uniformly glowing referees' reports, was the fact that you were awarded the 2014 Prime Minister's McDiarmid Emerging Scientist Prize. So if you're good enough for the Prime Minister, you may be good enough for Otago. So congratulations on that. So enough said on that, I think you get it. But before I ask Fiona to introduce you, a little bit more about why a Rowheath Trust Award and a Carl Smith Medal. Now, one is for the money and the other is for the glory, quite simply. The Rowheath Trust, established in 1964, or was established in 1964 to support the work of the university by Carl Smith, a prominent Dunedin businessman and a member of the University Council. And it was used to endow a $5,000 research support award that bears the Trust's name. So provided the audience agrees, you will, or you may have already received that financial inducement. At the same time, the university commissioned a medal in Carl's honour as the glory to accompany the medal. the money, I'm sorry. And so it gives the university a very good opportunity to recognise the contribution Sir Karl and his wife, Lady Catherine, made over many years after coming to New Zealand from the family estate of Rowheath in Birmingham. The family connection to the university and the award continues with Karl's daughter having lived in the Otago region and having been a regular attendee at these awards up until very recently. And it was a real pleasure in the first years of these awards to have Catherine come and present the award. Now, as well as the medal, there's also a very special little surprise as well, that Sir Karl was a well-known author in addition to his business and community contributions. In addition to his book Sweet Success, The History of Chocolate Maker Cadbury Fry Hudson, published in 1968, he was the managing director there. He was also known for his quirky and humorous tome, New Zealand from N to Z, that has become somewhat of a collector's piece, and in fact, I think that we may have a copy of which to give to you tonight, which is actually to Gladys with Best Witches from Maud in 1948, but we actually scour the second-hand bookshops to make this an even more special presentation for you. So you can decide which is preferable, the money, the medal, or the book. Accompanying to the medal, we have that book for you, and it will, I hope, find a very special place in your home alongside the medal for you and your whānau to remember this special occasion. So that's enough from me. Without further ado, I'd like to hand over to Fiona as Karl's head of department to introduce you in your talk. Nō reira, tēnā koutou, tēnā koutou, a tēnā koutou katoa.
Kia ora katoa. It's my pleasure to give a little bit of background to Karl. So Karl, so it's going to be a bit of a biography, sort of weaving in some of his great scientific discoveries, I guess, and leading up to introducing his talk. So Karl is actually an Otago graduate. He graduated with 2 degrees from Otago in 2002. A BSc in physiology, and also a Bachelor of Physical Education with honours. And he then moved to the University of Calgary in the cold part of Canada to complete his Masters and PhD, and both of those were in the neuroscience field. Karl worked with Professor Jadip Baines, and he studied a group of neurons that secrete 2 really important hormones, oxytocin and vasopressin. And Professor Baines, who was one of the referees for this nomination, says that Karl employed innovative techniques, including electrophysiology, to measure the electrical firing of these brain cells, as well as imaging to study changes in signalling pathways in these cells. Now, the quality of this work was recognised by the award of the Governor-General's Gold Medal, and that's the top award for a PhD thesis at the University of Calgary, which is a well-known and very prestigious institution. After his PhD, Karl, I guess, looked around, and he found that in physiology, we had Professor Alan Herbison working here, and Karl took up a postdoctoral fellowship with Alan. So that was associated with our department and also the Centre for Neuroendocrinology, and that was in 2010. Karl moved his interest to the main neural circuit that was studied in the Herbison lab, and that's the neural circuit that controls fertility. And he studied a group of neurons that secrete another hormone, GnRH. And working with a team in Alan's lab, Karl discovered this neuronal structure that Richard referred to, and it was just unknown. No one had recognised it was there before, so it hadn't been described. And it's the structure on a neuron that combines the properties of both an axon and a dendrite, and they named that the dendron. So this work was published in 2013 in the Journal of Neuroscience, and that's a prestigious journal in that field. And I understand at the moment it's sitting on a very impressive 89 citations. And Professor Herbison says that this work has fundamentally changed thinking on how the neural circuit controlling fertility operates. And now virtually every wiring diagram in this field now incorporates the concept of this dendron. And in 2014, Karl was awarded the Prime Minister's McDiamond Emerging Scientist Prize based on this work. Also in 2014, we appointed Karl as a lecturer into the Department of Physiology, and this is when he started to develop his own independent research program, and he moved to a third neural circuit, and that's the circuit that controls our stress responses. Now, since 2014, Karl has been awarded a string of continuous funding for this project, including, again, a prestigious Sir Charles Hercus Health Research Fellowship, a Marsden Fast Start, a full Marsden Project Grant, and most recently a Health Research Council Project Grant, all as Principal Investigator. Now, Karl is known for developing techniques that push forward our understanding of neuronal circuitry, and in particular, more recently, he's pioneered the use of fibre photometry at Otago. And this is a new technique right at the forefront of his field, and it allows specific populations of neurons to be monitored and recorded in real time in conscious behaving animals. It's kind of the ultimate of finding out what's happening in real time in neurons in the brain. And his first major paper using this technique to study neurons involved in the stress response has just been accepted into Nature Communications. So now I'd like to invite Karl to present his lecture titled Neural Signatures of Stress, and we look forward to hearing a bit more about that work.
So thank you, first of all, for the lovely introduction that I've received, and thank you for this great honour in being awarded this the Rowheath Trust Award and the Carl Smith Medal. So I think success in science is determined not just by the hard work of the individual, but there's a huge team behind those people as well. And I want to thank a few of those people before I start. So I want to thank my research mentors over the years, so my PhD supervisor JD, my postdoc supervisor Alan Herbison, and all the academic members in my department which have provided support since I started as a lecturer. I also want to thank all the support staff in our department that do all the kind of behind the scenes work that also allows me to do the research. They do a fantastic job in our Department of Physiology. I also work in the Centre for Neuroendocrinology, which is really a hub for research in neuroscience at Otago, especially in kind of the type of research that I do. And it's a truly world-leading centre for research, and I'm very honoured to be a part of that. I also want to extend a huge thanks to my lab, who are the guys at the coalface doing the research, doing the experiments on a day-to-day basis now. I'm going to be talking about the research that they do tonight. I can cover only a part of it because there's so many cool projects going on, but I just want to thank them for all the hard work that they do. It's much appreciated. And finally, I want to thank my family, many of who are here tonight, my parents, my wife Jo and daughter Sylvie, and my son Enzo, who couldn't be here, for all their support over the years as well. Okay, so now I want to move on, I guess, to the science side of things, and tell you about the research that my lab is interested in. I guess the research question my lab is interested in, and that is really trying to understand the brain circuits that control our stress responses. More, I guess, specifically, I'm really interested in the neural signatures, and what I mean by this is that the patterns of neural activity in the brain, the unique patterns of activity which ultimately drive behaviour, drive kind of physiological output. And I'll explain this more as I go through. So, as you all know, we have a remarkably complicated brain with billions and billions of neurons in it, with trillions of connections between all these different neurons. And we have dedicated circuits in our brain which do different jobs in our body. I'm really interested in the brain circuits, the neural circuits down here in a deep part of the brain called the hypothalamus. And as has been introduced tonight, I've studied several different parts of these circuits, these survival circuits that control water balance or fertility. But I'm going to tell you about stress tonight. So in this region of the hypothalamus, there are different parts. And I'm really interested in this one part here, this quite a beautiful structure called the paraventricular nucleus of the hypothalamus. So it's quite a mouthful. The paraventricular means it's on either side of this thing here. This is a ventricle. This is where cerebral spinal fluid goes through. And it kind of looks like a heart shape or a butterfly shaped structure. And these glowing things here are brain cells which have been labeled with a particular dye or marker. And you can see three different colours, three different types of brain cells that do different jobs in the structure. There are these green oxytocin neurons, there are these blue vasopressin neurons. But today I'm going to talk specifically about these orange neurons, these orange brain cells, which are the CRH neurons, the corticotropin-releasing hormone neurons. So these orange cells make this particular peptide called corticotropin-releasing hormone, but I'm just going to call it CRH for short because it's too much of a mouthful. So these neurons, I like to think of these cells as the master controllers of our stress response. They are the primary neurons in our brain controlling our neuroendocrine stress response. So when we encounter a danger in our environment, a potential threat, information is transferred from other neural circuits in the brain to these cells and tells them to become activated. Once they're activated, they release signals into the blood, which activate the pituitary, and then subsequent signals which activate our adrenal glands. So our adrenal glands are these structures on top of our kidneys on either side of our body here, and our adrenal glands release other hormones. They release stress hormones into our body. And in humans, the primary steroid stress hormone is cortisol. You may have heard of this before, people talking about stress and cortisol. So cortisol is our primary steroid stress hormone. In other animals, you may have heard of corticosterone. And cortisol, it travels throughout the body and it mediates many of the effects of stress. It acts on lots of different parts of the body to change its function and it helps us adapt and survive a stress or a threat. So it acts in all these systems here to change function. It acts in the bone, in the reproductive tracts, it acts in immune cells, it acts in mobilizing fuel stores, glucose, acts in the gastrointestinal system, the cardiovascular system, and importantly on the brain. And overall, it's changing all the function of these different organs in our body to make us more likely to be able to survive that impending danger, that impending potential injury that could result from this threat that's in our environment. So a lot of people think that stress is bad, but actually acute activation of this system, this pathway, is a good thing. You know, these systems have evolved to help us survive. And so when we have encounter a danger, a threat, acute release of stress hormones into our body are beneficial. They drive adaptations that help us survive the threat or the danger. When the system breaks down, when stress can become a bad thing, when stress hormones can become a bad thing, is when it's chronic. So either chronic activation of these stress circuits or chronic elevations in cortisol levels in our body are really detrimental to health. And that's because they change all of these structures, they cause long-term changes which end up leading to kind of disease states or predisposing us to disease states. Likewise, this can all start in the brain as well. So if we have for some reason, the neural circuits in our brain are not, the patterns of activity are not working right, and we have dysfunctional stress responses that start in the brain, this can also lead to diseases as well. Most notably, neurological diseases, depression, anxiety, post-traumatic stress disorder are all associated with abnormal functioning of stress circuits in our brain. So it becomes quite obvious that this is an important thing to study. We need to understand how the system works in healthy individuals. We need to understand how it works in response to acute stress, to understand normal function before we can understand and treat kind of the diseases that result from abnormal functioning of this circuit. Okay, so now let's look at the kind of profiles of stress hormone release into our body. So we can measure stress hormones in our blood, we can measure this in animals, we can measure it in humans. I'm going to just, use a human as an example here. The first thing to point out is actually that our stress hormone levels are never zero. So as you guys are sitting in the audience here, you have an ongoing secretion of stress hormones into your body. It fluctuates across time, across the day. And this is actually really important for your normal kind of function. Your normal body function, because this cortisol is important for regulating a lot of systems. Okay, so now let's imagine a hypothetical situation where you might encounter a lion. So say you're on safari in Africa and you're wandering around outside your tent and a lion jumps out at you. Okay, so this stimulus could rip you limb from limb, right? So it's a real threat, it's a real danger to you. This is something that you're going to want to mount a stress response to. So this information is passed through various neural circuits to your CRH neurons, which we would assume become activated and drive the activation and the release of cortisol into our body. So there's a massive release of cortisol. This is going to mobilize fuel stores and do other things to help us escape and, potentially fight off this lion if we have to. But hopefully it doesn't come to that. Anyway, the lion actually walks away, and as that stressor is removed, as the threat is removed, our cortisol levels drop back down towards baseline levels. So you're talking to the gamekeeper that night, you say you've encountered this lion, and he says, oh, don't worry, it's a tame lion. Actually, he's quite friendly, you can pat him, you can feed him, it's fine. And you're like, oh, okay. So the next day you're out again walking around and you come across the same lion, and this time you think, well, yeah, I trust that guy, but I'm not sure. You still have quite a large stress response, but it's smaller than the day before. The following day, you guess what I'm going to say, you come across the same lion. This time other people in your party are patting it. Someone's taking a selfie with it. It seems actually quite a nice lion, and your stress response is much smaller. You adapt over time, and this is actually a good thing. You want to only respond to stresses that are really dangerous, because you don't want to release too much stress hormone into your body all the time. You don't want to keep on activating these stress circuits, because ultimately that's damaging if you do it too much. So your body has systems in place to adapt to stresses that are non-harmful. And this adaptation is a normal process, it's an important process, and we know from stress hormone measurements that it takes place. What we don't know about is really what happens up here in the brain. What are the actual patterns of neural activity that drive, number one, what are the patterns of activity that drive this basal secretion? What are the patterns that drive this massive surge of secretion? And how does plasticity happen in the brain to drive this adaptation to these non-harmful threats? So I guess that's where neuroscience comes in. That's where my interest lies, is understanding the changes in neural activity in the brain which underlie the secretion of stress hormones into our blood. And that's what I'm going to tell you about today. So I want to understand, I guess, this regulation of this process at multiple levels. So I want to understand it at the level of a single brain cell, because that's the single brain cell, I guess, is the single unit of computation in the brain. That's kind of the unit that's doing the processing, and I want to understand how stress signals are encoded at the level of a single cell, connections of brain cells that are talking to one another, relaying information to one another, all the way up to whole animals where you have an organism which is behaving in its environment, encountering danger in its environment, trying to understand how neural activity changes behavior and endocrine stress hormone output. And then I guess the ultimate goal of the research is to translate this into better understanding stress responses in humans and diseases in humans. So today, I guess I'll tell you about the progress we've made really understanding stress from the level of a single neuron up to whole animals. We haven't quite made it to humans. That's more, I guess, a goal for the future. So for the non-neuroscientists in the audience, I just want to take a step back and tell you a little bit how a brain cell works. So this is a cartoon of a brain cell that you might see in a standard kind of textbook. And there are three different parts to it. Well, there's more than that, but three, I guess, simple parts to it. We have the central region, which is the soma. This is kind of where the genetic material is stored, where a lot of protein synthesis is done. Off the soma, you have these branches, which are the dendrite. The dendrite is actually the region that receives all the input from other cells. So all other cells that are making connections onto this neuron generally form on this dendrite. Information is transferred from one cell to another here. So information seems to flow from inputs onto the dendrite, through the soma, and then down this long wire, which is called the axon. And this transfers information from this cell and ultimately down to downstream neurons that it connects with. So you may know that the brain is electrical, that we have electrical signals in our brain, electrical patterns of activity, and these individual brain cells are also electrical entities as well. They process information and communicate with one another via electrical chemical signals. So electrical signals originate up here, but can then be propagated down this axon to downstream neurons. Importantly, it's actually the pattern of electrical activity which is incredibly important. So here is an example of a simple pattern of electrical activity in a single brain cell. We have three spikes of electrical activity, then a rest, 3 spikes of electrical activity and a rest, 3 spikes. So each of these clusters of electrical activity are called a burst of activity. So here we have 3 bursts of electrical activity, and it could be in this neuron that these bursts of electrical activity encode information. Something's happened at these 3 time points, and that neuron is responding to those things. So very simply, this is the neural code. This is how neurons kind of work. They encode information with patterns of electrical activity. Those patterns of electrical activity encode either things happening in the environment or they drive responses of the neuron, depending whether it's a sensory neuron receiving input from the external environment or maybe a motor neuron which is directly controlling the output of some circuit. So now let's look at a CRH neuron. Remember, these are the stress neurons that are controlling stress responses. They're a little bit different in that rather than talking to downstream neurons, they actually release signals into the bloodstream. So if we look at the pattern of release of these stress signals into the bloodstream, you see that after every one of these electrical spikes, you have a little blip of release, such that after these bursts, you get a burst of secretion that matches up with every burst of electrical activity. So from this we can see, hopefully you can see, that by understanding the patterns of electrical activity, we can start to see how neural activity is shaping stress hormone release in the body. So the important message here is that patterns of electrical activity in these stress neurons directly determine the patterns of secretion of stress hormones into our blood. So if we want to understand how stress circuits work in the brain, we need to understand the patterns of activity. So the next question is, how on earth do we record these patterns of activity? We want to record them from the level of a single brain cell, we want to record them in networks of brain cells, and we want to record these patterns of activity in whole animals as well that are running around and behaving and interacting with their environment. So how do we use a single technique to do all of those 3 things? Well, I guess there's a few different ways we can do it, but the way we've done it is we're using light to record neural activity. So I want to spend a couple minutes explaining how we can do this. And weirdly, it all starts with these green jellyfish. So these green jellyfish glow green because they make something called a green fluorescent protein. And some very smart scientists, they took the genetic information encoding this green fluorescent protein and they put it into other cells in other animals. And they figured that they could make other cells glow green. Some other smart scientists around the world, they changed the sequence for that genetic code for that green fluorescent protein, and they made it glow different brightness depending on the activity of the cell. So now we can basically take the genetic code for this green fluorescent protein and put it in a brain cell, and the amount of light that comes out of the brain cell is going to change depending on the activity of the brain cell. So it's like having a light bulb inside the brain cell that changes light intensity depending on neural activity. So with increases of electrical activity in a brain cell, we have increases in green light. And with reductions in neural activity, we have reductions in green light. So now by measuring just light, we can either measure activity from a single brain cell, lots of brain cells, and ultimately even in behaving animals, because we can measure light in all these situations. So we're going to use genetic tricks to get this green fluorescent protein into our CRH neuron. The modified version is called GCAMP. And now, by measuring light coming out of our stress neuron, we can reconstruct the neural code. So in this cell here that was firing these bursts of electrical signals, if we measure the amount of green light coming out of this brain cell, we can see that after each blip in the electrical signal, we see a blip in the green light. So now we can measure light as a proxy for neural activity and reconstruct the neural code in these stress neurons. So we can collect the light several different ways. We can use big chunky microscopes. So this is an example of the most impressive chunky microscope in the lab. It has lots of mirrors and lasers. And so you can take some nice photos. But importantly, we can put brain tissue underneath this microscope, and we can keep the brain cells alive in this wee tissue bath underneath the microscope by putting in solution that has glucose and salt and oxygen. And we can then study those individual brain cells, or those small networks of brain cells that are connected with one another. We can collect the light using these things here with wires in them. We can actually record the electrical signals at the same time as well. Okay, so this is what it looks like when we take a movie underneath the microscope. So this here is a single brain cell. This is the soma of the brain cell. These things coming off here are the dendrites I told you about before. And you can see that it's flashing on and off. So each of these flashes is a burst of electrical activity. So by measuring light, now we can see in real time before our eyes, even just looking down the microscope, we can see this happening. We can see the neural code of this particular neuron in real time. And I guess the neat thing is we can also see this in many different neurons. So if we zoom out, we can now start to see this flashing and correlate this activity between other cells. We can see if the activity is coordinated between cells, is it synchronized, what happens if we block a type of communication, does that block the synchronization? So we can do a lot of interesting things. We can do a lot of measures using just light. Okay, so the first person in my lab who really started doing this was Julia Gauss, who was at first an honours student, then went on to do a master's and is now in med school. And she was interested in studying the patterns of activity in these individual brain cells to see what they looked like, because we didn't really have a very good idea. So she measured the light coming out of these CRH neurons that were glowing green, and what she found was in most of the cells that she looked at, these are just 4 different examples, but she really studied hundreds of different CRH neurons. She found that actually the levels of light coming out as an indicator of neural activity was pretty boring, it was pretty flat, nothing was going on. And so we wondered, well, this may be a bit of an artifact of how we're doing the experiment. We've got a bit of brain tissue in a dish, and this doesn't really mimic the real life situation. In real life, you're going to have a whole brain with an animal that's getting sensory input from its environment, and we've really cut all that off when we have some brain tissue in a dish. But can we mimic that by, I guess, artificially stimulating the cell with a signal that it might receive in the whole brain? And the signal we used to try and stimulate these stress neurons was a signal called noradrenaline. So you may have heard of this before. It's a neurotransmitter which is released into your brain when you experience stress. So there are other populations of brain cells which are activated by threat and stress, and they send information to the CRH neurons by releasing noradrenaline. So we wondered if this neurotransmitter in the brain could change the activity of these CRH cells. And when Julia applied this neurotransmitter to the CRH neurons, she saw a really remarkable shift in the activity. They went from being very quiet to showing a really robust, massive increase in excitability. Not only that, they switched into this really unique pattern of activity, often characterized by these recurrent bursts of electrical and calcium waves. So Julia did a lot of work characterizing the receptors and the intracellular pathways, and I don't really have time to go into it, but this was our first insight into the patterns of neural activity in a single CRH neuron. The caveat being that it's in a dish under a microscope. So, with that caveat in mind, we wanted to start thinking of other ways that we could measure light coming out of the brain, now of animals as they interact in their environment, as they encounter danger and encounter potential threats. So we can use powerful microscopes to study single cells or small networks of cells, but we can't really plonk a microscope this big on top of a small animal's head. We need to have something more subtle, more delicate way to measure light coming out of a behaving animal. So we switched to a technique that Fiona touched on in the introduction called fibre photometry. So this technique allows us to use fibre optics to measure light coming out of an animal's brain. So when we first started doing this about 4 years ago, well, we started thinking about doing it, there wasn't any companies that were selling the equipment we needed. So we had to build our own, but we didn't really have the expertise to build our own, so we collaborated with some smart people at a local Dunedin company, Jesse Teat, Mark Butler and Campbell Young, who designed the electronics, designed the software to allow us to measure very small changes in light in an animal's brain. June Kim in my lab collaborated with Sue Hahn and Alan Herbison really to translate the system they had built into the lab and actually get real data out of it. So this is one of the first systems that we made. Basically, it has some light-emitting diodes in here that generate light, which pass light out through a filter cube. We have a system of collecting light that goes to a photodetector through an analog to digital converter to a computer. And what this allows us to do essentially is to shine a little bit of light into the brain of an animal and then collect the light that comes out. Importantly, we're collecting green light here, and I told you before that we can use this green fluorescent protein approach to measure neural activity. So increases in neural activity correlate with increases in green light. And this system basically does very minute measurements of the light coming out and allows us to reconstruct the patterns of neural activity. So I guess we were interested in many, many questions, because this technique really opened up a huge number of things that we could now study. The first thing we wanted to study was just the patterns of activity in these brain circuits under the resting state. So as I told you earlier, as you're sitting here, your stress hormone levels are fluctuating, and they show this kind of characteristic fluctuation over the day, even if there's no stress, even if you're completely calm. And we wanted to understand, or we wanted to know, do the CRH neurons in your brain also fluctuate their activity patterns across the day, even if there's no stress? So this was the PhD project of Caroline Fock, and she measured the neural activity from 8 a.m. in the morning to 8 p.m. at night, and then 8 a.m. the following day. So 24 hours of neural activity recordings from just the CRH population. And what she saw was pretty amazing, actually. She saw these amazing rhythms of activation in this neural network. Even though the animal is in a completely calm, resting state, we see these bursts of neural activity happening roughly every hour. So the cool thing about this is that we also know that in humans and in many species of animals, your secretion of stress hormone also happens roughly once an hour as well. So what we think is that these bursts of activity drive the ongoing rhythms and stress hormone release over the normal course of the day. She also found that this activity appeared to be higher at nighttime. So in rodents, rodents are active at night, and so it seems like there's higher activity in this circuit at nighttime when the animal's active. So that was a really cool first insight into kind of the basal activity patterns of this circuit without any kind of stress around. But when you think of stress, obviously you're thinking about an acute stress response. So we were wondering how the circuit changes when you encounter stress in your environment. So this was a project of June Kim, a postdoctoral fellow. He again recorded the baseline levels of neural excitability while there was no stress, no threat, and he saw the same kind of thing, ongoing kind of low levels of neural activity. But then he played a loud white noise to the animal, and immediately you see that the stress circuit massively switches on. There's a very robust increase in neural activity. But as soon as the noise switches off, the activity returns back to baseline levels. So what he found was that, similar to Caroline, there's an ongoing low level of activity in the resting state. However, as soon as the animal detects a potential threat in the environment, remember this is just a loud noise, it's not a real, it's not going to harm the animal actually, but the animal thinks it's a potential threat, there's a switching on of this system. If the threat is removed, the neural network switches off again. So this is really important. You don't want to have this system going on all the time. You don't want it active all the time. When the threat is removed, it's a good idea to turn off the system again. So what we've able to achieve so far with this fibre photometry technique is we've been able to start to understand the patterns of activity in the basal state, the resting state, which drive the resting secretion of stress hormones. We've started to understand how encountering a threat in our environment can drive a massive secretion of stress hormone, and when that threat's removed, the neural activity seems to switch off just as quickly. But you remember I told you earlier that also there's this adaptation over time, such that as you learn that something is non-harmful, it's advantageous to switch off these stress circuits. And this white noise, this loud noise that June used as a threat actually is the perfect stimulus to use, because if you present this over and over again, animals and humans quickly learn that actually it's non-harmful, it's not threatening, you no longer are stressed by it. So we used this to test what happened to activity or plasticity in this neural network over time. Yeah, so what happens to neural circuit activity in these CRH neurons as an organism learns that a potential threat is non-harmful? So this is the very large increase in neural activity on the presentation of the first time of this loud white noise. The second time on day 2, so this is only the second time it's been presented, the neural activity appears to be slightly smaller. On day 3, it seems smaller again, and day 4, it's even smaller. So if we just shade in the area under this curve to represent the total sum of neural activity in response to this potential threat stimulus, this loud white noise, we see that over multiple presentations, the neural response gets smaller and smaller. So what you see here is the real-time process of learning. The animal's learning that this is non-harmful, and it's shutting off its stress. It's not even activating its stress response because there's no point in activating it. It's actually beneficial not to activate it. So these increases in neural activity are driving stress hormone release from our adrenal gland. And I told you earlier that these stress hormones can also, they travel throughout our body, but they can also get back into our brain to change brain function. So there's a lot of previous research looking at the effects of stress hormones on how it shapes our brain function. And it's been shown previously that stress hormones change our ability to form memories, change our mood, our sleep patterns, but really interesting, they can also change our subsequent stress responses, make us more or less responsive to stress. So we're really interested in this idea, how are stress hormones actually acting on these CRH neurons to change their activity to potentially modulate our responses to stress. So now we move back to our microscope. We want to study single CRH neurons now, the single brain cells. Again, we have some tissue in a dish under this microscope. This time these brain cells have been labelled with a red dye, so each of these red circles is an individual CRH neuron. We can now, with these wires, we can record electrical activity from just one cell, and the one cell is just shown here. We can fill it with dyes and actually see the full extent of its morphology. But the important thing we wanted to do was to record the electrical activity and see how electrical activity changed with or without stress hormones around. So this was the electrical activity we saw that in these CRH neurons when there weren't any stress hormones around, when these brain cells had not been exposed to any elevations in stress hormone level. And this was the pattern of activity we saw if there was stress hormones around, or if the brain cell had been exposed to stress hormones. And you'll be able to see that the number of electrical spikes is less here. And we did a number of experiments, and this showed that actually these stress hormones were dampening down the activity of these neurons. So it seemed like when the stress hormones were feeding back on the brain, they would seem to be shutting down this neural circuit and reducing the activity. So, in summary, what I've showed you today is that we have a dedicated population of neurons in our brain, a neural circuit, which involves these CRH neurons, these corticotropin-releasing hormone neurons. And these neurons are really the master controllers of our neuroendocrine stress axis. So our lab in particular has been able to discover for the first time the patterns of neural activity that happen over the day and the non-stress state, just all the time, which are likely driving our ongoing rhythms of stress hormone secretion. We've also been able to show how stress actually leads to a change in activity pattern of these CRH neurons when an animal encounters a threat. There's an immediate strong activation of these brain cells. And this was going to release a lot of CRH into the blood and cause the large amount of secretion of cortisol, corticosterone from the adrenal glands. We've also shown that there are important kind of, safety mechanisms whereby this neural circuit can learn over time, it can adapt over time, and it shuts off its activity, gets less and less as an animal learns that a threat is non-harmful. And this is a good thing. We want to have systems in place to prevent excessive stress hormone release and prevent excessive activation of these stress circuits. And then finally, what we've shown is that once released into the blood, these stress hormones can actually feed back, travel back into the brain and act on these same cells here, these CRH neurons, to dampen down the individual excitability of these brain cells. Again, another safety mechanism to feed back and try and prevent excessive activation of stress pathways, try and prevent excessive release of stress hormones into our blood. So we think we're starting to make some progress into understanding, I guess, how these circuits work in a healthy brain. But the real challenge, I think, still lies ahead in understanding really what goes wrong in disease states. You know, what happens to these neural circuits to drive malfunction, to drive abnormal activity patterns which are associated with particularly we're interested in mental health disorders where you have abnormal stress circuit function. So that's a big challenge for the future. So I just want to wrap up by again acknowledging all the people that made scientific contributions here, all the members of the lab, the institutes that I'm a part of, the collaborators, all the many funding bodies, and finally thank you for coming along and listening to this today.
Wonderful. Don't sit down, Karl. We've got a few more words to say and a few things to give you. It's my job to give a vote of thanks and to summarise. As a physicist, the main thing I would like to summarise and impose on you all is that no good research is done without some optics. So everything circles back to physics and light, so thank you for making that point so eloquently, Karl. But it's also wonderful to see that in your research you've already, as an early career researcher, developed the ability to get a group alongside you, encourage and lead and motivate people to address really big questions. And it's also really great to see that whilst you have made outstanding progress so far, that there are many questions for you still to be stimulated with in your journey to come. I've also found that I've had one of my key mantras reinforced about stress and fear, and that fear is your friend, or a little bit of it can be as well, and that stress hormones are not too bad. Also, that we use strange language as scientists at times, and I liked very much that you use many acronyms along the way, GFPs and CRHs and other things. That's good, but probably the first time that I've heard a lion being treated as a stimulus. So careful, children, there's a stimulus over there. Avoid the stimulus. So I thought that was quite amusing. And clearly great that you have not only encouraged and engaged with academic people, but you've identified that in Dunedin we've got wonderful innovators and innovative companies that you can join with to help and develop the tools and techniques for you. So this part of the evening is the test. So if you all think that Karl has done sufficiently well to be awarded a little book and a medal and some other things, please join me in thanking Karl again for a wonderful talk. So I'll give you a couple of things and ask you to take a quick look at them, probably not open one, and then put them down. I'll give you a special something that is I think your lecture poster that you can keep as a memento, and of course this wonderful laminated version of From N to Z, a humorous survey of New Zealand by C.V. Smith. Thank you. And now I will ask the Vice-Chancellor to come and present you with the Karl Smith Medal. Vice-Chancellor.